Hey, say again. Oh, yeah, sure.
Oh, yeah.
Hey, say again. Oh, yeah, sure.
Oh, yeah.
Hey, say again. Oh, yeah, sure.
Yes.
Hey! Yeah. Wait. Do I wave? Can people see me? I'm waving if you can't see me.
I can't see you yet.
Oh, okay. Well, just know that I am waving, those of you out there who are waiting with bated breath.
Oh, by the way, I was muted that entire time too. So now I've got to. Man, I'm telling you, man, I really need somebody producing this stuff. Has nobody heard a damn thing I just said? It's okay. Wow. All right. Well, I tell you what, I'm gonna fix it so they can see you guys. And while I'm
Yeah,
doing that,
sure.
tell them who you are, Erica, and what we're gonna be talking
Yeah.
about. You do
Oh,
the
man.
introduction.
Yeah. Absolutely. All right. My name is Erica. I go by Gutsick Gibbon here on YouTube, which hindsight is 2020 with the username, but I've grown accustomed to it, and now I'm quite a fan of it. It's grown on me. Anyways, yeah, I got my undergraduate degree in animal science, and I picked up a minor in biology and in anthropology along the way. I got to go on this excellent trip to Tanzania in 2015 that kind of sparked my interest in primates and in human evolution in general. And so after I finished my degree, I was like, you know what? Why don't I just go for the big money and be a primatologist? So I went ahead.
What is that?
That's where it is these days. I'm telling you.
What's her name? Goodall?
Yeah, Jane Goodall.
Jenna Goodall, yeah.
Diane Fossey, the whole gang. And so I went ahead and got involved in a Masters of Research program over in London. And that's kind of what I'm doing now. I'm supposed to be in Malaysia right now doing my data collection. I was supposed to leave on the 18th of March, but in case those of you out there are living under a rock, there's a little pandemic going on, which has thrown something of a monkey wrench, pun intended, into the whole situation. I was supposed to be doing my collection on behavioral ecology of pig-tailed macaques, which was going to be really cool. But instead, I thought, why don't I just use this time to read papers and hang out on YouTube with the people that I like quite a bit. So
I'm glad you mentioned that because I forgot. Because of the whole pandemic thing, I wanted to because it's a science show, I wanted to thank Trump I've got my cure right here. I'm knocking myself out here, so I figure that whole thing should take care of it, and I'll be safe and good to go, right?
Yeah, the oral drinking of the bleach is good, but I've heard it's even better if you just go for the straight injection.
Straight in, like mainline it or something.
Yeah, absolutely.
Yeah. By the way, don't drink bleach, people. Don't put needles in you and put disinfectants. That's stupid. If you're going to use bleach, it's fine to disinfect water, but the ratio is 8 drops to 1 gallon. I used to do that on board ship and put sodium hypochlorite into water, which essentially. But yeah, you don't drink bleach. That's just stupid. Anyways, we also got Ruhif in here. Ruhif?
Hello. How you going?
How you been, man?
Good. Isolating for the last month or so.
Yeah, been there.
Yeah. Oh, like I said, I mean, the meme that you see that says, everyone's isolating, and then it goes introverts. I've been preparing for this my whole life, and I'm like, yeah, that's good.
Yeah. Yeah, it's been good in a lot of ways. You can indulge I've been indulging some hobbies that I normally haven't really had the time to do. So that's nice. I've been gardening quite a bit, which, I've never been good at, but I've managed to grow some plants this year. So,
Very good.
Awesome.
Obviously I would have been thriving in the agricultural revolution of humankind.
Mesopotamia.
So start us off, Erica, because I know you wanted a full day to kind of get back into this.
Yeah, man. I mean, and that's the thing. All right, so here's the lowdown for those of you out there new to the subject. So with human evolution, and indeed with sort of comparative genomics in general, what we do is we look at full genomes to compare, species A to species B and tell, how related they are. This is done within species as well. You can do it with humans with paternity tests. You can do it with your dog to figure out, what kind of mix it is. And it's a pretty surefire way of determining, ancestry. So kind of along those same lines, back in the day, there was a prediction made by some very intelligent evolutionary biologists who were essentially like, well, it's quite interesting because as everyone, including creationists such as Carolus Linnaeus, father of taxonomy, noted, it does very much seem like humans are related to the rest of the hominoids. But we have 23 pairs of chromosomes rather than the typical 24. So what's up with that? And so they were like, all right, well, one of two things has probably happened. Either there's been a fusion event, right, or the ancestral population had 24, and then there was sort of a loss for the rest of the hominoids, the gorillas, orangs, and chimps and bonobos. And so the prediction was like, well, it's probably more likely that a fusion happened because losing entire chromosomes isn't typically looked fondly upon. And so a couple of years later, right, we finally get the technology to get a closer look. And what we find is that chimpanzee chromosomes 2A and 2B are fused together in humans to create chromosome 2. And this isn't just, the way that we know this isn't just because obviously 2A and 2B in a chimpanzee are almost superimposable on human chromosome 2 once they're stacked on top of each other, but also because in the middle of human chromosome 2, we have a telomere fusion site, right? And then we also have 2 centromeres, and we have subtelomeric regions, all of which are just a dead freaking ringer for a fusion event. And the cool thing about this, as Ruhif has noted in many of his blogs and works, this has happened in other organisms as well. It's happened in pigs and it's happened in some of the equids. So this is pretty open and shut as far as genomics and comparative genomics and genetics in general goes. Except creationists don't like it very much. And so Dr. Jeffrey Tomkins, who actually has degrees from conventional universities, has decided to lead the vanguard of creationists into the future with his claim that, no, it's not a fusion site. And he has a whole host of bad reasons for why it isn't, even though you're hard-pressed to find someone who would agree with them. So, Ruh, if you want to go into, I don't want to hog and monologue too much here. Do you want to go into some of the...
we got hours. This is great. Take all the time you need.
I don't have hours.
Go ahead.
But like I just mentioned in the chat, it's been many years since I've looked at it. Like Steve would say, I've been hitting flurpers the last couple of years.
I know, man. You gotta stop picking up on flurpers, man. At this point, you're just being mean and bullying me.
it is more fun.
Low-hanging fruit.
That's true as well. So I'm quite happy with Erica monologuing as long as possible, and I'll chime in when I can. But I don't anticipate that to be much because, like I said, it's been a couple of years. Rational Mind is in the chat. I do want to point people.
It's been a while.
I do want to point people to his blog post that he made on Bayes' theorem and why it means that the fusion is actually positive evidence for evolution as opposed to just not evidence for it.
And by the way, that's something that actually I disagreed with Roof on, believe it. Originally, we had talked about this topic years ago, and Rational Mind did a brilliant blog on it. And before he did, though, I actually disagreed with Ruhif. And by the way, you're allowed to do that, even though I love the guy. I literally am in love with the man, if I in that way. Not like that, but I mean, he's one of the greatest guys I've ever met. And I disagree with him. And you know what? You're allowed to do that. I did think that the human chromosome site was positive evidence. I didn't take it from a Bayesian approach, which completely works. And I think Rational Mind is spot on. His stuff is unassailable, I think. But I think to break it down for the people that are not familiar with the topic, we need to maybe kind of put it very basic. And I think Erica did a wonderful job. But let me just touch on that a bit. So if I remember correctly, because it's been a few years, back in the early 80s, they had at least known that there was a fusion, but they only recognized it from the fact that they had it done by, what do they call it when you stain it? And
fish. Yeah, fish.
Fluorescent in situ. So they knew that it existed, right? They could tell that it existed, but they didn't find the cryptic centromere, because for a fusion to occur, you would have to have two centromeres, one of the actual chromosome, and two the cryptic or the inactive one. And at the time, they didn't have the technology to really sequence the human chromosome. They couldn't tell that if it really was, in fact, pancroglottite 11 and 12 or chimp 2 and A and 2B, right? Is that true?
Yes.
I understand. Okay.
Yeah. So they compared the fish stainings, and they look pretty similar. So it was good enough evidence.
There's a similarity there, because as Erica pointed out, we have diploid number 46, and all other members today have 8. And if we're related to primates, if we have common ancestry to all these primates, then why is it we're missing some chromosomes? What's up with that? That's not normal, right? But it is, as Ruhiv pointed out, because he had done some vast research on this. It happens quite frequently in the Munchak deer.
Yep.
And amoebas and some other things have these translocations. And I believe, if I'm not mistaken, this actual fusion site was a Robertsonian translocation, right?
In humans, no. Yeah, my definition of a Robertsonian translocation is that it must involve a centromere. So it's either centromere to centromere or centromere to telomere.
Does it? Because I don't remember that being the case. I thought it had to do which chromosome it dealt with, because this was because of a telomere fusion, right?
It's telomere fusion is what I...
In the humans.
Yeah.
But as to a Robertsonian, the common Robertsonians that occur in humans are pretty much in the chromosomes that have a short arm with garbage in it. So pretty much a short arm entirely made of repeats. And that's why people can survive okay with a Robertsonian translocation, because they don't actually lose anything.
Information just moved around.
Yeah, they do lose it. They do lose an arm.
Well, not always. No,
and they survive.
Do they always have to lose some information on Robertsonia? I thought that you could...
No, they can switch around as well. That qualifies as Robertsonian as well. But some people actually lose an arm. An arm of a chromosome, not an arm.
The short arm of the chromosome.
Yeah, and they live. So, gotcha. Yeah. It depends which chromosome, obviously.
I think it's important to note, and Jackson Wheat covered this in his video. He touched on it on Human Chromosome 2. But additionally, it's important to keep in mind that in primates, there are numerous, genera that have variations in the chromosome number. I mean, in gibbons, in many of the Macaca genus and Papio, you see variation in individual species that they freaking share a genera. So it's not like this is unusual.
Oh, true. And there's even primates that can interbreed with different chromosome number as well.
Yep.
I love the fact that you brought that up, because we're going to get into Tomkins in a second. And I apologize for interrupting you.
No, please.
The fact that you bring up Gibbons is this very big point, because if you go read Tomkins' work, he actually slides in a Gibbon, tries to put it under the great apes, and says, "Oh, look, this Gibbon has 50." And it's like, wait a minute, no, it's access, and they don't. And he doesn't say it's a Gibbon. He just says it's an ape, which but it's a Gibbon. It's not a member of Hominidae. It's a member of Hybloibatatae. How you pronounce it?
Yeah, the Hylobatids.
Yeah. So he sneaks that in there, and I even pointed that out. And I was like, wait a minute here. This is comparing apples to oranges now. So I thought that was really. But anyways, so go ahead and kind of tell us what Tomkins' argument actually is, why he thinks that this is not a fusion, why he thinks there's an active gene there.
Oh, well, I mean, God, that's the thing. Which argument by Tomkins are we going to go over? Because his style here with Human Chromosome 2 is throw everything to the wall and see what sticks. And individuals like Ruhif here have gone through them fairly meticulously and been like, look, this is a reason as to why this isn't a fair, assessment of the chromosome 2 fusion. I mean, one of the ones that is kind of classic, right, that he likes to propose is that there's a functional gene on the fusion site, which is the one that everyone really likes to dogpile on him for, rightfully so, which is the DDX11L2 gene. So the interesting thing is about this particular guy is that it's a part of a family of genes that are all telomere specific, right?
Yeah,
so they're all found.
Family. Yep.
Yeah, so they're all found capping the ends of chromosomes. So what the heck is it doing in the middle of human chromosome 2?
It's the only one that is, if I remember.
Yes.
Yeah.
Right. Yes, it is. It's the only member that is. And interestingly enough, the actual gene itself isn't actually spanning the proposed fusion site, but sort of like a ghost of it is, right, under some renditions of the sequence, right? And the interesting thing is that even that sort of ghostly reflection of the DDX11L2 gene has a promoter that's kind of off to the side, which suggests that it arrived there post-fusion. Again, this isn't something that isn't found anywhere else, which is something that I think Tomkins likes to do, is he likes to act as if there's this big spotlight on the human fusion, that this is, oh, look, it's so abnormal, it's so unusual, look at all of these strange things about it that prove that it's just not what we're saying it is. Completely, by the way, leaving out one of the most important facts about the fusion, which is the synotony between the top half of human chromosome 2 and the bottom half and chimp 2A and 2B.
Oh, it's huge. Yeah, it's a huge relationship.
That's I mean, he just doesn't even address that, throws it out the window and dunks it into the garbage can.
I think he actually changes a little bit. Ruhif, correct me if I'm wrong, but when he was doing some comparative, his graphic was highly exaggerated.
Yeah, he was working his graphics off the GEMSA staining pictures, and then claiming, I think Human Chromosome 2 was some enormously high percentage longer than the combined chimp chromosomes 2A and 2B. When you look at the actual DNA sequence in base pairs, the number of base pairs, it was very, very close. So he definitely exaggerated that, but that's his style.
Well, and a note on that, so there's Tomkins is also notorious for his most recent 84% comparison of the human and chimp genome. And while I was kind of digging into that, which I was coincidentally doing quite recently, I kind of stumbled onto some cool papers on hominid duplication events. Hominoid duplication events. And the interesting thing is the fact that the human chromosome 2 is a bit bigger than the chimp 2A and 2B is expected. Mm-hmm. You've got a divergence time of 6 to 7 million years ago, and to expect that after, because obviously the proposed sequence of events in human evolution is that the divergence happened prior to the fusion event. So it would be absurd to propose that the divergence happened and then boom, right after that fusion event, without any duplication along that 2A in the meantime. It's not realistic. You wouldn't propose that under any other lineage for any other organism. And yet here Tomkins is proposing that having a slightly longer portion in the human chromosome 2 is so out of the ordinary. Wow, how can you even compare them?
Yeah, and it's kind of odd because, like I said, he does ignore the synony, but the biggest thing, and did I get that right? Synteny, I think it actually is.
Synteny,
yeah.
Synteny.
Yeah. He ignores that, but the biggest thing that I notice is the way he does this. Okay, and you're right, there's a promoter region there. People agree with that. But the way he's reading it is in the reverse direction of a transcript. That there's a canonical from, and again, I'm going back years on this. I have not looked at this in a while. I know you've brushed up, but if I remember correctly, there's a canonical read, and then there's a reverse transcript that he's trying to say. But the canonical read is spurious just reads. It's basically remember the number is 3.9 RPKM from the testes, which is where they took the highest amount of read, but it's just spurious. And he's trying to say, look, there's some kind of canonical read here, and he's saying the gene is in the reverse transcript direction of the promoter, but that's what he's saying is an active. And so when we had people ask him, you're saying this is an active gene, right? And you're right. From what I remember, the DDX11L means, what is it? A type of a DDX. It's in the family of, right? Is it
L kind of refers to pseudogene in that thing. So that there's actually the DDX actual gene that does actually produce a protein. So there's a whole family of DDX genes. Of the family of DDX genes, there is DDX11, and that is a protein coding functional gene. Then somehow this gene got copied close to a telomere. It became non-functional, so it's a pseudogene. So that's a DDX11L. So the L signifies that it's non-protein coding, and then that DDX11L family has propagated throughout all the telomeres. So there's several layers or several hierarchies of how nothing this gene really is.
So how is he saying that this is an active gene there? What? Because I've looked. Remember back in the day, I was looking at Ensemble, and I was looking at all these data, and I couldn't find a single case where this was considered to be an active gene because there's no actual reads from it.
Yeah, if I remember. Oh, sorry, Ruhiv. You can go ahead.
There's no actual protein produced from it. So when he says it's in the reverse complement, yes, it is read in the reverse direction. That's fine. There are 2 transcripts for the DDX11L2 gene. So a transcript is just a reading from DNA into RNA, and then sometimes, depending whether it's a functional gene, gets translated into a protein. This does not. Generally, RNA versus protein, you kind of distinguish proteins as the more highly functional things. RNAs can be functional, but generally they are less functional and less important.
So when he says it's a protein gene, he's saying that this is actually encoding. So you have the messenger RNA that's actually being synthesized into some kind of valued protein, protein that's doing something. Yeah. Is that what he's arguing?
Yeah. Okay. Well, he's not actually arguing that, but generally, yes, proteins are more important. They do things.
Okay, but this doesn't have an encoding for protein.
No, but RNAs can do things. No one has yet proposed what this RNA might do. They've just said that it's transcribed, it exists. That's about as far as it goes.
Yeah.
So there are 2 transcripts that are produced from DDX11L2. One of them is a longer transcript that does actually span the fusion site. The fusion site is not important. Like the fusion site's an intron. It's actually removed when it's transcribed, or if it was. Yeah, ignore that. And then there's the shorter one. The shorter one is closest to the canonical version of DDX11L family.
Actually, you know what? Explain to people the difference between exon and intron when you do post RNA and you're trying to do when you're actually doing splicing. How does it? What actually is kept and what's not? Because maybe people want.
Yeah, so exons are the actual protein coding parts of a transcript, and introns are the bits that are dropped out and don't actually form part of the protein when it's translated.
A quick super chat. Stupid Horror Energy says for $5. Why is the fusion site so degenerate if it happened 6 million years ago? This is a great question because it is a degenerate region. Yeah, it's highly degenerate.
Who wants that one? I'm
Yes.
happy to do it. You want to, Erica?
Oh, I mean, I think it's as simple as to say it's a long ass period of time. I mean, to even suggest that you're going to have a completely intact fusion site after 7 million years, particularly like we said in an organism, the hominids being quite particular with this, especially back 7 million years ago. But I'm sure you guys know, like the hominid slowdown hypothesis, where we've got the mutation rate is slowing down, which is kind of cool. But it used to be quite a bit faster. So that degeneration would have happened relatively quickly, actually, right after that fusion occurred.
But why? Okay, so why is that his argument, though? This is one thing I never did understand. Why Tomkins is using the degeneracy of the fusion site of especially the cryptic centromere. Why he's using that as an argument against it being a fusion? That makes no sense to me. Isn't it evidence that
degeneracy? It's a bit of a red herring, actually. So obviously, a pristine telomere sequence is TTA, GGG, just repeated perfectly. Right. If you had 6 million years worth of mutation on that, then it should be about 98.5% identical. Right. Because that's our rough mutation rate, 1.5% from chimps in 6 million years. What Tomkins appears not to know is that if you look at the DNA sequence, not of chromosome 2, but of every other chromosome, what you get is, genes, and then on the end, you have this pristine telomere sequence. But between those two, you have the degenerate telomere sequence. So it's not pristine. It's actually composed of different telomere sequences as well. So TT, GGGG. So there's all these different motifs in there. They are degenerate in chromosomes that are not chromosome 2. So that's just indicative of the structure generally, in that it goes genes, degenerate telomeres, pristine telomeres that are regenerated during cell reproduction.
Which is indicative of what you would expect, though, from a telomere fusion, would it not be?
Yeah, I mean that's the whole hypothesis, is that
that's what confuses me. I don't know why he thinks it's not.
Yeah, the whole hypothesis
really trying to
is
understand his position.
telomere.
I don't get it. Why he thinks it's not, and that's what we would expect.
Yeah, because what you have is you got the genes, degenerate telomeres. This is on a normal chromosome. Genes, degenerate telomere sequences, and then pristine end cap telomeric sequences.
You want them to be pristine, so you avoid having those telomeric fusions, right?
Yeah, and that's the pristine ones is what causes the whole loop that actually caps off the chromosome.
Yeah. I don't know the process, but there's a name for that.
Yeah, I forget it all.
We got Rational Mind. He can remind us of that.
Yeah, Rational Mind can get in. Actually, he'd do a much better job than I would.
You want me to go back in my biology 20 years? Good luck with that. I remember. There's a body. There's the whole mechanism. It's called a what is it? It's like a whole complex that does this.
Yeah, Shelterin complex. Yeah.
Was it the
Shelterin complex?
Shelterin complex. There you go.
Yeah.
This is bringing back some memories here, man.
Wow. Same. The hypothesis is that the telomere machinery was broken in some way, and because of the cell replication problem, the chromosomes shorten, and if the telomerase is not adding new telomeric repeats on the end, then they will shorten back to the point where they no longer protect the chromosomes from fusion. So you've got the genes and the degenerate telomeric repeats, but no actual telomeres. So they are susceptible to fusion, and that's when they fuse.
So this happens every time a cell when a cell goes through meiosis, it splits, right? And it only gets split a certain amount of times because of the Hayflick limit. But each time telomerase is supposed to come in and build up that telomere again. Mm-hmm. And if it does it, then the shorter the telomere, the more likely of a fusion.
Yeah, so there's a replication enzyme called polymerase, and it replicates the DNA. When it gets to the end, the machinery fails a little, and it shortens it, basically. It doesn't copy the whole thing. So telomeres naturally shorten, and it's telomerase's job to come along and lengthen them so that they then curve around and perform and provide that end capping protection. So the hypothesis is that telomerase machinery failed in some way. Your chromosomes got shorter, back to the point where there was no pristine telomeres on the end. You just had the degenerate ones that are there on every other chromosome. So you've got degenerate telomeres on one chromosome, happened to another chromosome, they joined, you've got degenerate telomeres in the middle. So it's not like they joined, they fused, and there were thousands of pristine telomeres sequences. They joined degenerate. So the expectation of or near pristine telomeres is a wrong, incorrect expectation.
Yeah, because we would expect if there's going to be a telomeric to telomeric fusion, it would be because there's a degeneracy, because they don't have the accurate amount of telomeres, right? There you go. This is why, again, Tomkins doesn't make sense to me.
It really doesn't. I had it explained to me once, like magnets, right? Magnets are inherently attracted to one another, but if you put a cloth in between two magnets, it's going to prevent them, from coming together and joining at their ends. And so you can think of those pristine telomeres as the cloth that prevents them from joining, and the tips of the magnets as being degenerate telomeres. So there would never be a scenario where the fusion would occur, at least to my knowledge, where the fusion would occur with pristine telomeres in the middle.
Correct. Yeah, it means if there were pristine telomere repeats there, it would mean that the telomeres weren't working for some other reason, which I cannot think of.
Well, let me ask you this. I've never really talked to Tomkins. I've never talked to him, definitely on air. I had some correspondence years ago, I think, with him.
We were going to visit him in Texas, remember?
Yeah, I know. You and I, Ruhiv actually comes out and visits me every so often. I mean, Ruhiv is actually in real life friend of mine who I adore. We go hang out in LA and tear up the town.
Drive cool cars.
But we were going to go actually visit Tomkins in Texas, and hopefully we still might after social distancing dies down. I would really like this.
Yes.
But from what I remember, Tomkins basically is running a lot of the same kind of positions that Dr. Humphreys has. Well, I do have correspondence. But Humphreys and Tomkins both have the position that this must be the way it is because it could be no other way, because they've denied science to the point where anything that just shows that their position is wrong must be wrong by fiat unconditionally, because it can be no other way. They've thrown away science, in my opinion, in lieu of their own dogmatic and ideological beliefs that the earth is young. And in order to keep that, in order to sustain that, they have to throw these things up there. Like Eric brilliantly said, throw whatever they can up there, see what sticks. Just throw it out there. And the people that don't want to learn these topics, the people that just blindly follow them, go, look, here's a PhD, Dr. Tomkins. He says this on Answers in Genesis, must be right. Those are the people that I think that Dr. Tomkins and Dr. Humphreys and on the PhDs in Answers in Genesis, they cater to. Because I got to tell you, I think at this point, after I've been doing this for a very long time, my channel started debunking Jung creationism. I think a lot of these people are just straight up dishonest.
Absolutely.
Straight up dishonest.
Yeah. I mean, and honestly, you can't. I hate to say this because I really try to give people the benefit of the doubt.
It's hard, I know.
I used to as well.
But you get to the point where you're like, all right, so you've got Jeffrey Tomkins, a geneticist, like he has a degree in this kind of thing, and he's
a PhD.
Exactly right, and he's going around making rudimentary mistakes. That my experience with genetics is I took it in my undergrad. And even I'm capable of reading his papers and going, wait a second, hold on here just a minute. And what's weird to me is like I said, I was looking at his genome comparison recently, and the way that he gets that 84 number in part is that he might look at a 300 base pairs, right? And let's say there's a nucleotide substitution right in the middle. So he might look at that and then say, okay, well, then the chimp genome and the human genome, despite that there's just one, base pair difference, they're only 50% similar. And so you look at that,
because the reason why he does that is because Ruhiv actually, because when Tomkins first put out his first, and I'm going to say a paper, but it really wasn't, right? Okay, when he put out, he did a genomic analysis, and he did it he came up with 70%, and as you guys both clearly remember, there was a bug and blast. So if you did batch, you would have a problem with it, right? And Ruhiv noted that, and Tomkins, to his credit, recognized that. And I think he couldn't help but not recognize that, but this was a known bug in blast. Okay, so he had to do something. This is where the dishonesty comes in, and I'm just going to throw it out as dishonesty. my principal charity is shot with him. But
Same.
let me throw it out there. So what I think he did was this, and dishonestly so. He did the genomic analysis again, okay, and then he changed the parameter to ungapped. So any indel that had multiple nucleotide differences in base pairs, instead of being counted as one, he counted each individual nucleotide difference as a difference, and then said, "Oh, look at these major differences." When it should be one event, because an indel is one event usually, right? And it might have different base pairs, but instead of having like 10 different base pairs, it's one change if you're going to compare them as a fair analysis. Is that something along those lines, Ruhif, that happened?
Yeah, I think my blog post, I think I put out the alphabet. So A through Z, or Z. Z to cater for your audience, Steve.
Us Americans,
so A through
Z. We don't know about you weird foreigner.
We're Z. We're Z. So you're comparing A through Z, and then you've got your DNA sequence that you're comparing it to, and it might have a couple of substitutions in it, like the D might be a K or something like that, and the J might be an L, whatever. But you've got substitutions, not indels, all right? So if you compare those, and you've got 2 substitutions out of 26 letters, then you're 11 divided by 13, you're 90% or whatever, 90% similar. What Tomkins did with this ungapped parameter is that if there was an indel, so an insertion or deletion relative to the other sequence, then setting this ungapped parameter in BLAST, it would actually stop the comparison. So he would be comparing a 26-letter sequence at the top. He'd be comparing a 27 or 25-letter sequence on the bottom because of the indel. So let's say it was an insertion, and he's comparing it to a 27-character sequence. And let's say that at the 20th character is where the indel is. He would configure BLAST to stop counting at 20. So he would only find a 20-character match against the 26-character comparison sequence. And that's what he would report. I explained that to him.
But doesn't that show he's willfully trying to manipulate it to get a number that he wants? The
fact that he didn't address it in his response to my paper and still used it. So my paper was, first of all, yes, there's a bug in BLAST, and that's where you got your majority of your result wrong. But you also used ungapped, and that shouldn't.
The second time, no, did he use ungapped the first time?
Yes, and the second time.
Oh, okay. I thought just the first time he didn't. I thought the second time he used the ungapped, but then changed it, like you said, to what he was looking for. So okay, so both times he did use ungapped. And he got the 70%, which is just ridiculous.
Yeah.
Well, and so the weird. I'm sorry to interrupt you.
No, go, please.
But I think this is important to kind of insert in here, which is that you'll notice this with creationists very specifically with genetics, is that there's no standardization across the animal kingdom. It's okay to use ungapped and get these wild numbers with humans and chimps, but the second that they're comparing, tigers and lions so that they can get their cat kind, their biblical cat kind, then the methodology is pristine. Then we're going conventional. Then we're using the classic methodology, which really to me is the point where I was like, all right, wait a second. There's no way this could be a mistake. It's not a mistake. It's got to be intentional. Yeah, which again, I really hate to do, but I don't see another way. I mean, without insulting Tomkins and being like, you're an idiot geneticist because you should know better.
Oh, sorry. To be fair, his PhD mentor was a creationist as well. So that's, caveat to that. I don't know who it was, but he had an interview with a creationist-friendly radio station podcast thing, and he did talk about his PhD mentor being a creationist and obviously sympathetic to his position.
We got a message to you, Ruhif. RJ, James Downer says to you, tell Ruhif we're happy to see him as a face, as Jackson and I cited his excellent post on Tomkins and Rocks, which is the book that they did.
Wow.
You've been cited, man. You've been cited by Downer and Wheat.
That's very cool. Then the other day, Conspiracy Cats.
Okay, great. But
Conspiracy Cats had a
I might actually
debate.
do something with Wheat. We are talking about writing something as well, but it's kind of similar to what he did with James, but only want to write something philosophical. He's writing, another book, I guess. He's asked me to kind of help with chapters. Hopefully I can actually be able to do that. I love Man, to say that I corresponded with Jackson Wheat would be a thrill, or even with RJ too. That would be a thrill. I mean,
Yeah.
but I don't know how
Awesome.
much he wants to do with philosophy. But yeah, I'm stoked about that.
Oh,
So,
I was like the other day,
get excited.
Conspiracy Cats had a debate with Standing for Truth, and I was like fangirling, and he's like using my shit, and I was like, holy shit, this is awesome.
Wait, Standing for Truth was using yours?
No, Conspiracy Cats.
Oh, Conspiracy Cats was.
Conspiracy Cats was.
Yeah, you know, big time on
actually,
that when they start using your stuff, right?
Yeah.
Yeah, Ruhif, you I was looking in the comments under one of your videos, and you were actually answering some questions for Standing for Truth. And I was like,
Yes.
saw some questions, and I was like, wait a second, standings here. I was
Yeah.
like, And so then I went to his channel, because that was about 4 months ago. This happened this
Still
morning, which
sane.
is why I remember.
Yes.
Well, and here's 2 months later, he releases the ultimate Human Chromosome 2 debunking video. It's 45 minutes long, and I was
I
like,
haven't seen it.
I had read
I've seen it.
who was it? Standing for Truth, and the other guy he does, Matt.
oh, Raw Matt,
Yeah.
yeah.
And I had tried reading, and I probably got through most of it. But there's this like, if they're calling.
Their book, yeah.
Oh. My will to live had diminished to the point where if I hadn't have stopped, I probably wouldn't be here to this day. It has so many errors, so many fundamentally, basically wrong stuff. And I'm like, and they're just putting it out there like, oh look, we're geniuses. And I'm like, holy cow.
If Standing for Truth's done a video on Chromosome 2, then I might do a response. He came onto my channel the other day about, I think it was more about orphan genes, and I haven't done a huge amount on it. But if he's done a 45-minute video on Chromosome 2, then I might have a crack.
Because I don't have the skill set to do the technical stuff against a lot of these young creationists. I realize I never really have. I facilitate the conversations, and I know enough about the topics to discuss them. But I'm not going to write a refutation against Tomkins. I'm not going to write a refutation against Humphreys. Right now, I would show myself. I was mentioned in the Journal of Creation by Humphreys, and I was mentioned in another philosophical journal in response to Humphrey's work by Dr. Lachelt, who is a PhD in geophysics or something, I believe it is, a really specific field, but he does solid state electronics that he writes for. And I'm not going to be able to like ever get to a point where I'm going to refute them like Ruhif can. So I love the fact that if you will look what it like Stand for Truth and then refute it, and we can talk about it together.
That'd be cool. I mean, like I said before at the start, I haven't looked at this stuff for 2 or 3 years, really. Like I did a couple of videos on it in 2018. I mean, flat Earth is dying, really.
It is.
It's still
because
fun.
You've killed it. Congrats.
Still fun, but I would like to do another genetics video. So I'll check out Standing for Truth.
We gotta get back on the science, because I mean I've been heavily into philosophy because I just been enjoying it more, and I think I'm better at actually refuting some philosophical arguments. Science, because I don't have the background in some of the stuff that you require to really do a comprehensive analysis to refute somebody's work. I think in philosophy it's a little bit easier in some regards to look and take apart a philosophical argument. It's just, well, I shouldn't say it's easier, it's different, right? It requires a different skill set, because it's certainly not easy. Philosophy is hard, like extremely hard, but it's a different skill set, and I'm finding myself migrating to that side, and that's why I would love to have Ruhif get back into this, do more of the science stuff, and then, hey, let's discuss it, because I know Standing for Truth doesn't know what the hell he's talking about.
Well, he's definitely one of the better.
Yeah, no, and he could be really good if you learned it properly, right?
Yeah.
Well, the thing, he's also, I've never had a bad interaction with Standing for Truth, and I've interacted with him quite a bit.
Yeah, no, I don't have anything against him personally. We get along fine, believe it or not.
Oh yeah, but I mean that's the thing. I mean, one of my biggies, and there is a chance that he will watch this and then make a response video to it. I hope you realize that.
Probably.
But yeah, I mean, those guys can put out videos. Let's put it that way. They're averaging like 44 a month or something insane like that.
Can you imagine if they actually educated people properly? How amazing they would be. But go ahead.
Yeah.
No, I know, but yeah, I mean, I almost think, and I would feel comfortable saying this too, Standing for Truth, that there's like a mental roadblock that gets put up the second he gets too close to siding with conventional science as far as evolution goes, because I had a talk with him on Modern Day Debate about creationist genetics, so to speak, and one thing that he really likes to push is the ENCODE project's human functionality numbers, which because ENCODE has 80% of the human genome, even though very similar to what Tomkins did with our
They changed
DX.
what a pseudogene actually is considered to be and functionality.
Well,
It's transcribed.
anything that's transcribed.
No, it's not even that.
Yeah.
It's more than that. If I remember correctly, in the encode, it wasn't just anything that's transcribed. It's anything that has any type of function. If it moved, if it had a conformational change of some kind, if it was a conformational change, it considered to be something along the lines of active, and so they would consider it in the encode project as at least active or non. Garbage or non.
And they call DDX11L2 important as well. And I'm like,
right.
Well, sorry. First of all, from the debate, but from the debate the other day, even Standing for Truth said, it could be that Adam and Eve were created without the fusion, and then during the bottleneck of the flood or whatever, they fused, and that's how they can explain it under a creationist thing. The problem with that is that there's this period from creation to the flood with no fusion and therefore no important DDX11L2 gene. So you can't have it both ways. You can't have an important gene and then claim that it may have happened later. Creationists must claim that it was created
from the beginning, created that way.
Yeah.
Well, here's the thing. I think that again, when these just like the scientists have to give up science when they have to hold on to this belief. Standing for Truth would be under the same thing. He has to hold on to this belief because of his position. Now, he might try this as an experiment. Say, look, I'm going to assume that young Earth creationism isn't true. And I want to learn it the way it's properly understood and then see where that gets him. Because, look, I'm willing to throw away my entire worldview and say, let's look at it from the young Earth creationist perspective. But every time I do that, it's like, this is just fundamentally wrong. I mean, it's just, even like Humphreys, his math was incorrect. And Dr. Lachelle had pointed out to him that his math was correct. I even brought it to Lana's attention. Everybody I brought it to was like, this is not the same type of graph. This is a linear, this is a bimodal. It had a totally different type of curve. And it's like, these are not dissimilar functions. And he eventually, by the way, did admit that his approximation was incorrect. However, he hasn't publicly come out with that yet, I don't think. But he has recognized that math is math, and no matter what he wants to put as the age of the Earth, when you're shown that your math dealing with natural logarithms is wrong, or at least nowhere near the approximation you want, if a curve's like this and you have a line like this, those are not approximations of each other. How to recognize it. There's some hope. But I think Standing for Truth
same with Tomkins.
Worldview isn't sustainable in light of the actual evidence. He did that.
Yeah, like Tomkins recognized that there was an actual bug in the software.
Right.
I'm not sure
objective.
why he hasn't recognized the ungapped problem.
Dishonesty.
Yeah. It really is wild, and that's the thing. I mean, to me, one of the biggies, right, was the fact, and this is something that I bring up because you mentioned the age of the Earth, so it was brought to my attention. But you can't get around the radioactive decay law. It's not something that you can move around without, appealing to miracles or accelerated decay. Which even in a lab, even with non-geologically relevant materials, they couldn't get the decay rate to accelerate above 1%. And the cool thing is they got the creationist brain trust on this. Like, they put RAID out there in 2005, and they were like, you guys, AIG was like, go and prove that radiometric dating is a boo-boo. Go and prove it. And they came back afterwards, and John Whitmore was like.
I love the fact you mentioned. That's a little bit more I like radioactivity, obviously, because that was what I did. But real quick, before I even touch on that, because that's a brilliant point you make, and I do want to talk about that. Rational Mind says that if a single transcript was produced, it functions according to the broad definition of ENCODE in 2012, and that's absolutely right.
Yes.
Correct me if I'm wrong. Even if it was one single spurious transmission or spurious transcript, it was considered to be functional, if it was spurious or not.
Yep.
And I'm like, that's not the canonical understanding of functionality, is it?
No, it was biochemical activity, which includes obviously transcription. And there was a paper by Dan Grauer, I think his name was, Graur, who went through a really rigorous mathematical explanation of why a truly functional component of the genome cannot be higher than about 25%.
Yeah, right.
So someone Google that and stick it in there.
That was 2017. I brought that up in my conversation with Standing for Truth,
Yes.
my most recent conversation, because I was like, look, you can't, because Stan's been going around and saying, the trajectory of the human genome is that we're going to keep finding more and more function. And I was like, no, look, 2017, there's a cap. And as we all know, with regard to science, right, you have to appeal to the most recent literature. Absolutely. Because they're dealing with the most recent data set. Now, if you want to reject the most recent literature, that's your prerogative. But then you have to provide a reason.
I totally agree.
Give me 1 second. I'll be back.
No problem. And by the way, whatever you type is going to be shown. So, but anyways, so go back to the RAPE project, because the work that I had facilitated with Dr. Lachelle and Dr. Hankey, when it came to Dr. Humphreys and the RAPE project, is the reason I was mentioned in the Journal of Creation. And I was also mentioned in Lachelle's paper for facilitating this, because this was really important. They were talking about the helium diffusion rate, right? But specifically, when I had asked Dr. Humphreys that very question that you had brought up about accelerated decay, because what they're trying to do, and this is called the heat problem, and this is a problem in young Earth creationism. They admit this. They say this is a problem by young Earth creationism has not been resolved yet. And I say, well, what is your evidence of such a thing called accelerated decay? Now, you astutely brought up there are certain types of things that will accelerate. 0.8 to 1% is electron capture, K capture. Mm-hmm. In certain types of isotopes where it has to do with your S shell, that it's more likely the case than some other isotopes where the electron in the innermost shell is going to be absorbed by a proton, right? But that's very rare, but it can happen. But that's not going to get them where they want to go. I mean, not even close. 0.8 or percent is very small. Okay, so it's not going to get them where they want to go. But I asked him, so what is your evidence for accelerated decay? And I'm not even exaggerating here. This was his answer, because it could be no other way. Because that's literally what he's saying. It has to exist. It's like, why does it have to exist? Well, the Earth is 6,000 years old, therefore it has to be the case of accelerated decay. I'm like, circular reasoning, man. Do you not grasp this, that you're literally doing circular reasoning and you have a PhD in physics?
Yeah, and the thing is that it really shows how much, and this is more with your layman creationists than the bigger guys, the alpha chads of the creationist world. But we're working with complex methods in geology and in physics. And so another thing that I hear quite often is, well, we don't know the amount of parent material to daughter material. And it's like, well, that's cool, but isochron dating can skip that whole part.
On any kind of isochron dating, you don't care about the parent to daughter relationship. You just need to have that line.
Yeah, exactly. Yeah. You don't need it. And so that, that's gotten rid of the two big bugaboos that Woodmarap likes to
I explain that all the time years ago because people relate that to carbon dating. And I'm like, look, isochron dating and carbon dating, not the same thing.
Yeah.
There's a completely different ways of dating stuff. And I explained exactly what you said. You don't have to know the initial conditions of the parent isotope if you're going to do isochron dating. What you're looking for is that nice line.
Yeah.
And if you have it linear. And by the way, those geochronological formulas dealing with Taylor series, I've looked into those and I've tried to explain those as well, but I kind of gave up. It's been too many years. I don't know if you know my particular background, but I used to train people to operate nuclear power plants.
Oh, wow.
I had to at least understand some basic stuff about radioactivity, right?
Right.
And so I when I was talking about the RAPE project, this exactly was kind of a little bit more in my wheelhouse. But look, this is not how it works, guys. You cannot, because you have to account for all that heat. You have to say, look, if you want to say that the Earth is 6,000 years old, and they say, okay, then they admit there's millions of years, and it's not really like billion, but it's actually hundreds of millions of years of radiological decay, heat, energy that they have to account for. Because if they were right, the Earth would be liquefied. It would be just way too hot. The mantle, there's so much energy there, because most of the energy heat from the Earth is actually from uranium and thorium decays.
Mm-hmm.
So imagine all the heat from uranium and thorium, instead of having a span of hundreds of millions of years, compactified into 6,000 years.
Oh yeah, I mean, and it's not just that, it's combined with the radioactive decay itself, which you're having to cram from 4.8 to 6,000. You're also dealing with all of the heat that they have to have for their crazy catastrophic plate tectonics in the Noachian deluge. I mean, and the crazy thing is, someone pointed this out. One of my buddies actually pointed this out to me. But at the Creation Museum, on their map, their flood map, they've got Rodinia. They don't even have Pangea. It's Rodinia, which means what we're dealing with is the movement of Rodinia to Pangea, and then Pangea to the modern sequence of continents, which means the continents in 1 year are like zooming all around at the speed of light, essentially, and the friction with them on top of the mantle and up against each other is adding an enormous amount of heat to the equation. I mean, like you said, we're talking liquefying the crust of the Earth.
Oh, tectonics
many
kills
times
them.
over.
That was the whole point years ago. There was a principle called catastrophism, which was back in the 60s, basically destroyed. But they thought that changes came across in catastrophic events, right? But then plate tectonics came along and explained phenomenon. That really harmed the creationist movement because they wanted catastrophes to be the case. Because look, global flood, upheaval, fountains of the deep. By the way, again, which we've explained to like Nephilim free. If you wanted to posit, there's a huge amount of water being spewed out in fountains. The enthalpy of that system would be so great, it would no longer be water, it would be steam, and it would, again, destroy the Earth. We call them everyone dies scenarios.
Yeah.
But they don't explain that. They just rationalize it away. They never actually explain, okay, give us the mathematics of this. If you ever asked Standing for Truth or Nephilim Free, show us the math involved in this of how you have superheated water at atmospheric pressure that's not superheated. I mean, that's not like in steam. It's not going to be in liquid state.
Yeah, I've brought this up many times in conversations with people on YouTube, but I used to have a coworker who was a young Earth creationist, and he was very much the type who was like proselytizing in the workplace and, things like that. So I was like, all right, look, I did a lot of this in school. How about we go have a coffee and chat it out? So we started getting coffee like once a month and going over this kind of thing. And the catastrophic plate tectonics and the heat issue was my big, mallet on top of the watermelon for him, because I was like, all right, look, when we ended our conversation, we were growing to an end. I was getting ready to leave for the UK, and I said, I need an answer for this. Like, you keep dragging your feet and telling me you're going to look into it, but you got to just tell me, like, because you're the guy who's coming around saying teach the controversy in schools, but your hypothesis is precluded. It's not just not supported. It's precluded by the data that we currently have. So you have to explain to me why you think it should be taught in schools if there's no actual model that works. And his answer stuck with me because that's why I bring it up so often. But he says, well, as far as I know, they're working on it.
Yeah.
And I said,
that's the RAID project, right? The
okay,
RAID project is still saying we haven't figured it out yet, but we will because it must be the case. Now, somebody pointed out in the live chat, Baumgartner has conceded that CPT, I think it's catastrophic plate tectonics, would not work without God's direct intervention. And by the way, he has a point there because I did a presentation, and Roof, do you got a split?
Oh, in a minute, yeah. Okay. Yep.
I did a presentation ages ago on polonium halos. You remember Gentry's work, Robert Gentry's work?
Oh, God, yes.
That's one of the very few times I felt comfortable doing a refutation, an analysis of why his work was wrong. And I did a PowerPoint presentation, just 2 hours long, explaining in great minutia why he was incorrect. Gentry hadn't seen it, but Gentry has said eventually, when people have pointed out why his stuff is bogus, that God, he invoked what's called singularities. And what he meant by singularities are one-time events where God intercedes and makes it the case. Thus, at that point, the polonium halos is now taken out of science and put into, whatever you want to put it into, right? The garbage bin.
Yeah, just invoking
because he couldn't explain, because the information against Gentry's work is vast. And it was very much, I loved explaining it. It was a very great presentation, I thought. But he had to admit, and I wasn't the one that brought it up to Gentry. I just worked on other people, consolidated it. But he admitted it, that, yes, this, if you look at this, the way that people are telling him it's science, yes, of course, you're right. This makes sense. But God did it this way by a singularity event or a miracle. And I'm like, now Gentry, you're not doing science anymore. Right? I mean, this is what happened with Baumgartner. By the way, I was going to write John Baumgartner recently and ask him about Humphrey's math, because Humphreys was insisting his math was correct. And Lachelle had asked me, hey, why don't you get in touch with Baumgartner and ask him what he thinks of it? So.
Well, and that's the thing that really kills me about this. I'm all for people, having their faith and whatever it is that they want to follow. And I'm very much actually okay with someone saying, look, I think this, whatever, that there's a big global flood, but I'm doing it on faith because that's what I think. That's just faith. I just think it is what it is. I'm going to chalk it up to being like, all right, whatever, you're going to believe it, you're going to believe.
Scientism, I'd call it.
Yeah. And but the problem, you're right, the problem arises when someone comes in and they say, but science supports it. It's creation science. And that's when you say, oh, really? Let's take a look at that model. Let's see precisely what it is that you're trying to propose here, and then analyze it under the lens of every other avenue of science. And if you've got guys like Gentry, going around and saying, all right, yeah, you're right, I'm going to have to appeal to miracles, or like what Woodmarap said, which is at the end of the Rate Project, he's like, we're going to have to assume exotic solutions will be found in the future.
Mm-hmm. Right. That, by the way, that's the last thing I think that the Rate Project had said, wasn't it?
Yeah,
in the Rate Project itself, right?
That was the concluding remark.
That was the conclusion.
Yeah.
You're exactly right.
Yeah, and if you're going to say that, cool. But then you got to put a disclaimer at the opening of the Creation Museum or the Ark Park where you're like, look, some of this we have a little bit of science for. And the rest of it, we've got some exotic solutions that are going to come in the future, and we've got some faith. Because you can't go around proposing it like it's a complete model when a solid, percentage of it requires
95% is bogus.
Yeah, requires you to just throw your hands up and say, well, someone else will figure it out in the future.
I do have to go. Yep. I have to go and sell my old house, believe it or not.
Oh wow!
Oh man, really?
Yeah.
I thought you just remodeled. I thought you moved, or I thought you remodeled your house.
No, this is a new house.
Yes.
And we moved in over a year ago. We left our old house empty for a year, just whatever.
Which one was the murder
home? This one.
This was the murder home.
Yeah. Left the other house empty for a year and finally got it renovated in the last few months. And now, because we're not allowed to have open houses or auctions here at the moment, or in-person auctions, so we're not going to go and spend freaking $100,000 on a real estate agent commission. So I am doing open houses, well, not open houses.
Yeah,
I'm doing by appointment inspections.
Cool.
Who was the? We're going to talk about that story one of these days. But who was it? Because there's a big story in Australia on this.
Yeah, I know, but it also gives away my address and whatever. So,
you mentioned it before, though.
True, but it's. I mean, with doxing, it's not difficult to find out people's addresses. It's just not something you just don't sort of
that makes sense.
Publish. Yeah. Anyway, this has been a great chat.
Yeah, appreciate it.
I'm actually a little bit excited now to, like you did say that Standing for Truth put out a video, so I'm going to go and watch that.
Yeah, the thumbnail for it, so you know what to look for. It's a Photoshop of a gorilla centaur, with the top half being a gorilla and the bottom half being like an elephant. I saw it and I was like, well, it did grab my attention.
Excellent. I'll go and check it out. Standing for Truth, if you're watching, hit me up on Skype. I'm actually very surprised I haven't been interrupted by 4 kids so far over the last hour.
Did you let them know that, hey, this is daddy time?
No, I just locked the door and
Hey, kids they'll find a way in, dude. They're resilient.
All good.
Yep.
All right. Anyway, thanks for the chat. I shall catch you guys later.
Great to meet you.
You too. Right. Thanks, Eric. Bye.
All right. This is going to mess up my Skype call. So anyways, let's get back to the whole DDX11L2, then we kind of wrap it up a little bit. I think you've kind of given away the crux of what Tomkins is saying. And then I don't know what, I mean, he just hasn't given up on it, and it's coming back around, I think, because a lot of people that I've talked to have started mentioning it again. So this is kind of why I wanted to do this topic, because I was like, wait a minute, if this is going to be brought around again, maybe we should deal with it again.
Oh yeah, absolutely. I mean that's really it. The crux of it is that Tomkins has put out this work suggesting that the reason why, amongst other things, right? There's another thing he says where that telomere fusions don't happen, that it's always satellite telomere fusion, or I think satellite.
Yeah.
Yeah. So those.
Where does he get that from? Why does he say it has to be a satellite or satellite telomere?
Honestly, I don't know, because and Ruhif did a video on this that I watched earlier today, but he found very easily, just by doing a quick search on PubMed, 2 papers covering telomere fusions in equids and in pigs, which blows my mind. And I've brought this up. This is something I also complain about very often. Some of these claims that PhDs creationists like Tomkins or Georgia Purdom does this sometimes too. They make these claims that a cursory Google search would have elucidated. You know what I mean? Their literature, and RJ Downard talks about this a lot. The source methods are very weak for even some of these PhDs. They just don't.
Well, I think she's dishonest too. I really do think. I know that like apology has done a lot. He has said. Now I haven't talked to her. I have no connections with her, but I remember she did a video ages ago, like 5 years ago, and I had put you remember back in the day when people actually you put a comment on and people actually responded to comments. You have big old long 500 comments later kind of thing. Okay.
Back the good old days.
Back in the good old days. I kind of missed that. I had 5 years ago. I had so many threads that hit 500 comments. It was ridiculous.
Right.
But one of them was on Georgina Perdom's video. I made a comment. I was like, look, she's wrong here, and I pointed. And of course, people just like on her side, and I'm like, but you're just repeating what she's saying, but you're not explaining why she's correct on this or why I'm wrong on these things. And I don't remember the exact details, but I think she's a legit PhD too, so she has to know. I think that she's just full of crap.
Well, and another reason for that, and I bring up talking points multiple times usually because they tend to keep being relevant. But they wheel Georgia Purdom out to talk about geology when Snelling's not around. To talk about like Noah's flood and all of these types of things. So the Answers in Genesis kind of crew just interswaps individuals, professionals, whatever, and it doesn't really matter what the topic is most of the time, which kind of says to me that there's very little application of what their expertise actually is, because it's only surface deep.
Yeah, and well, like somebody had mentioned in the live chat, Vibrant Brantley said that Georgina and them, they want it for the money. I don't think that. I don't think they do it for the money. I think that Georgina is a true believer. I think Humphreys is a true believer. I seen his correspondence. I really am convinced he 100% believes what he believes. However, that presents a problem when you're faced with science that disagrees with that, because you have a cognitive dissonance then. And I think that Georgina is in that state. I think that she understands obviously the material, but her belief, her wanting to believe this supersedes anything else. Right? Yeah. So I don't think it's for the money. I think that she is a true believer, but in order to sustain a position that has been disproven, you have to be intellectually dishonest. Because her position is disproven. It's just not a matter of discussion or debate. Young Earth creationism is disproved fundamentally across the board by multiple different disciplines. For her to hold on to that belief, she can do this. But to do so, you got to be intellectually dishonest. And I think that comes out of people lying, but they justify it either A, they know they're lying and they're doing it for the good of good, lying for Jesus, or they're lying, but they're really not even aware that they're lying. I think it's possible. Right. They don't recognize what they're doing is lying, and people go, well, that's not really lying. It is a form of intellectual dishonesty, right? It's not.
Well, but it's not malicious necessarily.
Not malicious,
right?
But there's a disconnect there in their brain, right?
Right. And I think another kind of support for that comes for the fact that they do so little peer review. I mean, they really do have their own little isolated community where they peer review each other's work. You got to sign the statement of faith to work there, and the pews are usually full. And at like the Creation Museum where they do like those live presentations or Answers News, whatever, they're stocked with people who are there not to challenge them but to support them.
Yeah, it's not peer review though. That's like, hey,
it's
support, and very rarely do they ever kind of like tell each other that they're wrong. Like my stuff isn't peer reviewed. Just because I have people review it and people that are experts review my blogs, it's not peer reviewed. But but unlike them, if somebody finds something wrong with what I write, they're going to tell me. I assure you, right? It happens. They're not going to probably tell each other, because I mean, there's no way somebody could have read Humphrey's work. If you go to my webpage, www.greatdebatecommunity.com, I have Humphrey's response, and in that response, there's some problems, and people that know about the topic in his side should have said, hey, dude, this is not right. This is mathematically incorrect that you're saying that this is this. But it took Dr. Lischeld, who is a Christian, by the way, but not a young Earth creationist, to point it out going, this is simple math. You screwed it up. This is objective. Let's talk about it. You know?
Yeah.
And nobody caught that when they read Humphrey's work, that on his side? Really? A math problem?
There's no way. You know, I have such a hard time with that too, because it's like, you know, like I said earlier, like, you really want to give people the benefit of the doubt, because I don't know, like, you just hope. I had a conversation with Aaron about this, but it's like when you are involved in science even tangentially, there's sort of this mutual agreement with people who are involved with science that the other person is coming at it from an angle of, let's find out what's true. You know, not what we want to be true, but what is. And it's difficult to get out of that mindset when you're talking to people who are bad faith actors or disingenuous, or even, like you mentioned earlier, like they're just in a different headspace where they've got, you know, their science box and their young earth creationism box. And that sucks, because it's like so many of these people could be doing some amazing work in conventional science. And the guys at BioLogos prove that you don't have to divorce from your faith to do good science. Francis Collins proves that too. So I don't understand why it's got to be so dogmatic. And what pisses me off too is that they go around acting like their interpretation is the authoritative interpretation. You know what I mean?
And I agree with you. And I like BioLogos. BioLogos had a blog, but
right.
That Wayne Fillmore, he wrote something, and it actually got picked up by BioLogos, and that was about his and I debate, because our debate. He left Young Earth Creations because of our debate, pretty much.
Oh, wow.
And that's pretty amazing. But BioLogos is an organization that I think does want to do active science. Now, people can criticize it all they want, but, you know, at least they're trying.
Sure, sure.
I'm going to be working with, this is a good segue, by the way, before we end this. I'm working with Reasons to Believe now. We're going to be doing a new show starting live in West Covina after all this pandemic stuff, but we're going to be doing it from home first. We got a promo today that we started cutting. It's going to be with Dr. Fuzz Rana, if you know who he is.
Yeah.
It's going to be him and I co-hosting. Hopefully we'll get some Dr. Hugh Rawson every so often, because he's part of RTB, and he's a really cool guy. I get a chance to talk to him frequently now. And so I think RTB is an organization that they do want to talk about science. Do I agree with them on their science? No. Let me make that very clear. I do not agree with Fuzz Rana on his science. I have his books. Like they gave. Look, I got all his latest books from. He gave me Human 2.0, and I've got Improbable Planet. Which, by the way, if all the books that I have that I've been able to read, Improbable Planet is actually pretty good. You might want to check.
Yeah, it's is that it's an ID for fine tuning? Yeah.
No, what is and it isn't. Yeah, I mean, most of the book is about astrophysics, and he's an astrophysicist, so it has to be about science. And I have yet to have anybody really say this science is messed up. I have Dr. John Kroon coming on here soon. He's an astrophysicist. Maybe I'll have him kind of check it out a little bit. But I didn't see anything substantially in error of the science. But then, of course, then he argues, yes, that hey, we have survived like 5 major extinction events, right? We are so unlikely for life to be here. So he is running that argument, right? Do I think it's a good argument? Not so much. But you know what's better than some I've seen is, like, you know, it is truly amazing that we are here when there has been so many extinction level events that have all led to us having the ability to apprehend our universe. Because if it wasn't for any one of those events, we could not be. Who would be apprehending on Earth these things? It probably wouldn't have happened. Now, again, go ahead.
Yeah, I was going to say, I mean, I'm doing a series on my channel right now on mass extinctions, and I've actually, if you like mass extinctions, you've got to absolutely, I'm going to shill for this book by Peter Brannon called The Ends of the World, which is an excellent book that covers all 5 of the big boys and some of the lesser known ones as well, sort of the Ediacaran and then your end cap Anthropocene, potentially. Hopefully not, but things are looking pretty grim. But yeah, the mass extinction thing is really, really fascinating, especially when you get into the nitty gritty of what actually went down. I mean, the Permian, for those of you who don't know, Russia just detonated. Like, you basically had flood basalts going into a coal seam from the Carboniferous, and the countryside just exploded, which is wild. And like you said, you look at it you step back and you look at it and you say, wow, it really is crazy that we're here. But I do think that you kind of have to keep yourself in check with that, because you fall in danger of a probability bias, right? Of being like, well, we're here, so it must have been meant to be.
It's an anthropic argument, though.
Yeah, I don't know either way. So I'm not making a claim one way or the other. But I do recommend, I should give you a paper to put into the link in the description, because there's this cool paper with regard to abiogenesis that I've kind of been reading into abiogenesis a little bit recently, because I'm supposed to have a talk with someone tomorrow about it. Actually, John Maddox on Standing for Truth's channel. So we'll see how that goes. But there's this really interesting paper by a physicist, actually, I believe, on entropy, and how entropy, right, and sort of this dispersion of heat is the goal. And it turns out that life is very, very good at that. So the proposal in so many words is basically like that if the conditions are right, life becomes inevitable, just because it's an inevitable cog on that train of entropy, which I thought was
I've seen something like that
great.
before, and there's a process called ratcheting. And ratcheting is where you have a localized decrease of entropy that allows for complexity that then increases the overall net entropy of the universe. So as long as the entropy goes up overall, everything's conserved, everything's hunky-dory. So I think that you're right. Life seems to be an inevitability, given what we know about chemistry and physics, relationship between physiochemical reactions. I think maybe life is somehow a kind of inevitability, which is the case. Then there should be a lot more life out there on other planets, and some of it should be intelligent, to be able, at least to be able to apprehend the universe. But at the same time, look. I would have to have you back. We'll have to have this discussion about anthropic principle. But look, we know some great stuff about science. Well, if aliens exist, what do they know about science? Do they formulate the same conclusions? Because they have to have the same laws of physics. Do they have electronics? What would be alien electronics compared to ours? There would have to be very similar. It's almost like, what would be so different from an alien science to what we have, based upon the same knowledge and understanding of the universe and the same fundamental constants.
Right? Yeah, it's a blast to speculate on.
I love that kind of stuff. But anyways, like I said, to wrap that portion up, though, I will be doing stuff with Reasons to Believe. We have a question out there now. What would you ask a Christian? It's on my Facebook page. You can also leave answers on here. That's going to be one of the things we start bringing up. The first episode is, what would you ask a Christian? What would you ask a non-believer? But I'm going to be the non-believer, obviously. But I'm going to be arguing my own arguments that a lot of atheists in the atheist activist community have just thrown me aside and said, oh, well, Steve's wrong. Well, RTB and other theists have taken me very seriously. They like my arguments, and I'm starting a show based upon look, let's have a discussion about these things, because I've been shut down by the atheist community by fiat. Steve's just wrong, ergo don't have anything to do with him, kind of thing, which I think is a very intellectually dishonest way to approach things. Matt Dillahunty does that. Arn has done that one. I love Arn, especially on his phylogeny stuff, but philosophically speaking, he has exercised me, and I think that's a little disingenuous. But anyways, I'm going to be doing this new show. Hopefully it's going to be huge, because they already get an audience of about 500. We're going to bring our audience from the Non Sequitur show, and we're going to be working with that. And it's going to be a combined effort to have theists or believers against non-believers, like I should say, believers and non-believers. Okay. And I think that's going to be amazing, because that's the dialogue that I want to have. I want to have the conversations, and I don't want to shut people down just because you disagree with them. Like even Standing for Truth, I have no personal thing against him. If he wants to have these conversations, let's have them. I do have issues with people that do make things personal. Why have a conversation with them, right?
Yeah, I agree. And I think, philosophy tends to be, I can grapple with it, but at an arm's length, just because I'm not nearly as good, or specifically I'm not nearly as well read as I probably could be. But I do love watching conversations between people, particularly who know what they're talking about, because the best kind of discussions where you can see both sides. I mean, everybody enjoys a good curb stomp, of course, in certain like flat Earth. That's why people watch curb stomping. Yeah, that's what I was going to say. That's why you watch flat Earth debates, right?
Yeah, exactly. There we go.
But the thing is that a good dialogue where you can kind of get multiple vantage points, I think, is always valuable, even if you accept nothing of what one side or the other says. But yeah, I that's just my
Well, do you
thoughts.
remember we were saying that, look, flat Earth is dying. One of the reasons they're dying is because we've addressed everything they had to say. They got nothing else. They're out of ammo.
Yeah.
They've got nothing, and everything has been shown that they're just fundamentally wrong. So they are dying on the vine. But I said that was going to happen eventually.
Yeah.
But you don't see flat earthers out there like they were. Now, you got some go on modern day debate every so often, but that's just spectacle. It's entertainment.
Yeah.
Nobody takes the flat earthers seriously in the community. They have no standing. It's because they've been shown to be wrong. And the only reason way to do that is by talking to them. I don't have problems with the conversations. I just want to have honest dialogue. And that is from the theist or from the non-believer. Honest dialogue is important. So I want to thank you, because these are the conversations I love having. This was science. This was informative. It wasn't too technical, but at the same time wasn't, base level conversation.
Oh yeah.
So I hope we can have more on that. So.
Yeah, I had a blast, and I'm always down for a conversation. I will monologue, especially if it has anything to do with primates. I will monologue your head off. I was nervous when Ruhif was coming on, because I was like, oh God, I watched some of his stuff to prepare for this. I was like, he's gonna know it all already.
Yeah, well, he's pretty knowledgeable. I wouldn't have known half the stuff I knew about the DDX11L2 and Tomkins' work had I not read Ruhif's.
Oh, he put the legwork in for sure.
So with that, guys, good night. Probably gonna have a rum and coke night this Saturday if you guys want. It's been a long time since I had one. Those are always fun. And Erica, you're welcome to drop by. We just sit there and drink and talk. I don't know, about any kind of stuff.
A dangerous game.
Right. So, anyways, leave comments in the video description. Share. If you guys got questions on the Tomkins thing, message Erica or myself. Leave comments. But I want to thank people for watching. Again, I noticed that my science videos don't get much traction. People either like either more the philosophical stuff or the drama type stuff. But I'm going to keep that on my other channel. I want to have this channel strictly for science and philosophy as much as possible. And it's unfortunate they don't watch a lot of the science stuff. I know a lot of my audience doesn't come here for the science, and it's unfortunate.
Yeah.
That's what I'm going to be putting out is more science. So if people don't like it, whatever, go do what you got to do, right?
Right.
So,
yeah, but it's there. It's available. That's the important part.
Exactly, and I think it's entertaining. Oh, by the way, Standing for the Truth is in the live chat. He says Roof is a nice guy.
Hey, Standing.
So, by the way, Standing Truth, the offer's still open. I know you said you're going to join us many times, but something always comes up. But email me, and maybe next week, you, me, Erica, and whoever, we'll have a round robin kind. I don't like the debate stuff. People say, will you debate Standing for Truth or Nephilim Free? No. Not only no, but hell's no. I like to have the discussions, and that's the difference between modern day debate.
Good with the discussions.
Yes, yeah, no, I think that he would be better at the discussions, right? Yeah, exactly. So, anyways, leave us with your shilling for your channel and all that stuff. Give me any links, and I'll put them in the link, video description later.
Yeah, absolutely. Yeah. Do I verbally shill, or do I just provide the links?
Both. Why not both?
Oh, okay, cool. All right. Come to my channel if you want. It's just Gutsick Gibbon. And I do a lot of videos on sort of busting creationism generally, cool anthropology stuff, mass extinctions, human evolution. It's the good stuff. If you like science and fun opening animated intros that are painstakingly hand drawn by yours truly, drop on by, leave me a comment. And that's it.
Death and destruction.
Yeah, absolutely.
Nice. All right, guys. Good night, and we'll see you next time.